Hello dear students, welcome to my channel Kaveri Science Tutorials. Today we are going to see class 10 science 2 chapter 1 heredity and evolution. In that the first important process of protein synthesis may be confusing for some students, but I have made it easier for your understanding. So please listen carefully and we will go step by step. What is mean by protein, what is mean by central dogma of protein synthesis, what is DNA strand is made up of. So these things we will see one by one here. You will be your any doubt will be clear in case of protein synthesis. So first, proteins are necessary for the proper functioning of the body, and in human beings, in bones, ossein protein is present, in blood hemoglobin is present, in we can say skin or hair melanin is present. So these antibodies are present in the blood. Hormones, enzymes, all are proteinaceous. So there is a very much importance of proteins in our proper functioning of the body. So how the cell is going, each and every cell of the body forms protein. Inside the cell, protein is formed or produced or synthesized. All is having same meaning. Protein is produced inside the cell. So which cell organelle is there which helps in the protein synthesis? Which is the site of protein synthesis in cell? So different cell organelles are there in the cytoplasm. So ribosome is a cell organelle in which proteins are synthesized by the different types of cells. So different types of proteins are synthesized by different types of cells, and that ribosome is called as site of protein synthesis, or it is a place of protein synthesis. Now what is meant by protein? For the formation of protein, what we have to form? For the formation of protein, we have to form the chain of amino acids. So proteins, if in examination, if the definition of protein is asked, so your answer should be proteins are defined as the chain of amino acids. So proteins are the chain of amino acids. So if the chain is formed, then the proteins are formed. So how this protein synthesis takes place? So in previous standards we have seen the structure of DNA and types of RNA, and both are involved in the process of protein synthesis. RNA आणि DNA दोघंही 3 टाइप्सचे RNA होते. मेसेंजर RNA, mRNA, राइबोसोमल RNA, rRNA, transfer RNA, tRNA. So all these three types of RNA is involved in the process of protein synthesis. Again, DNA is a genetic material in all living organisms, but here in protein synthesis, only unwinding one strand is used in the process of protein synthesis. So the strand of DNA is made up of nucleotides. In this way, the chain of nucleotide is present on the one strand of DNA. Another strand also contains chain of nucleotide like this. Now we have seen the structure of nucleotide. So the nucleotide is made up of sugar, pentose sugar, nitrogen base, and a phosphate group. So what are the types of? So this is called as nucleotide. So the chain of nucleotide is present on the strand of DNA, and nitrogen bases are present, adenine A, thymine T, G, guanine, and cytosine C. So these types of 4 nitrogen bases are present. Instead of RNA, instead of thymine, uracil is present. So here we can see, if there is a pairing between adenine and thymine, guanine and cytosine in this way, then they are said to be complementary to each other. पेअरिंग जर होत असेल तर त्यांना आपण कॉम्प्लिमेंटरी आहे असं म्हणू शकतो. तर कॉम्प्लिमेंटरी हा वर्ड ज्यावेळेस त्यांचं पेअरिंग प्रॉपर असेल, adenine pairs with thymine, guanine pairs with cytosine, then the bases are said to be complementary to each other. आता now how the process starts? 3 मेजर स्टेप्स आहेत प्रोटीन सिंथेसिसच्या. फर्स्ट आहे ट्रान्सक्रिप्शन, सेकंड आहे ट्रान्सलेशन, आणि थर्ड आहे ट्रान्सलोकेशन. ह्या तिन्ही प्रोसेसेसचे डेफिनेशन बऱ्याच वेळा बोर्ड एक्झामला विचारलेल्या आहेत. सो लिसन केअरफुली. फर्स्ट स्टेप, आता त्याआधी आपण सेंट्रल डोग्मा म्हणजे काय ते बघूया. सो DNA with the help of RNA performs the function of formation of protein. So protein is synthesized by DNA with the help of RNA is called as central dogma of protein synthesis. So after that we will go to the first process of protein synthesis, that is transcription. ट्रान्सक्रिप्टचं मिनिंग आहे कॉपी. ट्रान्सक्रिप्ट मीन्स कॉपी. तर काय कॉपी करायचं आहे? So the information present on DNA is the information in the form of nucleotide and nitrogen bases present on the nucleotide. So this information is the information for the synthesis of protein. So this information is copied on mRNA. So one strand and the nucleotide is present like this. So this is the structure of mRNA, messenger RNA. So the information present on DNA is copied on mRNA for the synthesis of mRNA, and mRNA is having complementary bases to the bases present on the DNA. So in this way this information is copied on mRNA for the synthesis of mRNA, and this process is called as transcription. So proper definition of transcription is the information present on DNA is copied on mRNA and for what purpose? For the synthesis of mRNA. mRNA is synthesized, therefore this process is called as transcription. Now the mRNA synthesized in the nucleus comes in the cytoplasm. हा एक सेल आहे, त्या सेल मध्ये न्यूक्लियस आहे. So DNA is present in the nucleus. So nucleus मध्ये काय होईल? mRNA synthesis होईल. Formation of mRNA will take place inside the nucleus. After synthesis of mRNA, it comes in the cytoplasm of the cell, and when the mRNA synthesized comes into the cytoplasm of the cell, it joins with the ribosome cell organelle for the process of protein synthesis. So the information or the code for now the mRNA is going to form the chain of amino acid. How? So the code for a particular amino acid is present in the form of a group of 3 nucleotide, and that group of 3 nucleotide is called as triplet codon. So who discovered this triplet codon? One scientist is there, and this scientist is of Indian origin, and name of the scientist is Dr. Hargovind Khurana. Discovered triplet codon, and therefore for his discovery he has got a Nobel Prize for the discovery of triplet codon. So in this way this is about the mRNA has thousands of triplet codon. So mRNA after synthesis comes in the cytoplasm, joins with the ribosome, and when it joins with the ribosome, suppose this is the first triplet codon AUG, and example we can take AUG codes for a methionine amino acid. AUG codes for methionine. So methionine is brought by tRNA. So this tRNA is transfer RNA. It carries amino acid. So this is the structure of tRNA. tRNA carries or brings amino acids for the synthesis of protein one by one, and these amino acids are according to the code present on mRNA. The tRNA brings amino acids. These amino acids are joined together by a peptide bond, and in this way the chain of amino acid is formed, and this process is called as translation. So the second process of protein synthesis, important process, is translation. Means tRNA brings translation. Definition, for proper definition, for example, tRNA brings amino acids in the ribosome, and there is a chain of amino acid is formed, and tRNA has anticodon complementary to the bases present on mRNA. That process is called as translation. Means it is called as second important stage is called as translation. Now the third step is translocation. Now the meaning of translocation: trans means change, location means position. Now we will see the definition of translocation. Definition बघूया. So during this process of chain formation of amino acids, now you can write like this: during this process of protein synthesis, ribosome keeps on moving from one end of mRNA to another end by a distance of a triplet codon. This is called as translocation. Ribosome changes its position from one end to another end. Means again we will see the definition of translocation. Trans means change, location means place. Ribosome, during this process of protein synthesis, ribosome keeps on moving from one end of mRNA to another end by a distance of a triplet codon, and this process is called as translocation. So in this way, many chains of amino acids are formed to form a complex protein. So we have formed the chain of amino acid, and by that we have produced synthesized proteins. So proteins are very important for the functioning of the body. Ribosome in the cell is a site of protein synthesis, and in this way, by these three processes, and DNA with the help of RNA and produces proteins in each cell. ह्याच्यामध्ये तो सायंटिस्ट डॉक्टर हरगोविंद खुराना डिस्कवर्ड ट्रिपलेट कोडॉन अँड ही हॅज गॉट नोबेल प्राइज. ते एक येऊ शकतं. प्रोटीनची डेफिनेशन एक येऊ शकते. ट्रान्स्क्रिप्शनची डेफिनेशन येऊ शकते. ट्रान्सलेशनची आपण प्रत्येक डेफिनेशन दोनदा घेतली आहे. तुम्ही प्रॉपरली लिसन करा. आणि थर्ड वन ट्रान्सलोकेशनची पण डेफिनेशन येऊ शकते. सो आय थिंक दॅट इट इज इझियर फॉर यू. इफ एनी डाऊट्स यू कॅन लिसन. परत पॉज करा, परत एकदा ऐका हा व्हिडिओ, मग तुम्हाला अजून क्लिअर होईल अबाउट द प्रोसेस ऑफ प्रोटीन सिंथेसिस. सो इन दिस वे जनरली जास्त तर यातल्या प्रोसेस पण येऊ शकते, बट आतापर्यंत जास्त डेफिनेशन आलेलं आहे. त्याच्यामुळं दिस क्वेश्चन इज फॉर टुडेज लेक्चर. इन नेक्स्ट लेक्चर वी विल सी अगेन. बाय. थँक यू.